I want to start by thanking the organizers for including me in this riveting day of talks. The program is filled with great speakers whose work I've been following for some time, so it's an honor to be included alongside them. So I'm going to talk about two studies that explore the relationship between gene expression gradients and the functional organization of the brain. I'm going to take both a local view in the hippocampus as well as a more global view across the whole brain. So I certainly don't need to introduce this topic too much, but we all know that the brain can be characterized in terms of regions representing areas of shared function or architecture, but also gradient-like representations where gradation and function is organized systematically across specific linear or nonlinear axes. So regions and gradients coexist and interact hierarchically to influence overall brain functional organization. So the origins of this organizational structure might come from patterns of gene expression during development. I'm showing two examples here, one from the developing spinal cord on top, and also an example from the cortex at the bottom. But in both cases, broad sort of gradient-like expression of a few genes help to coordinate and recruit the expression of other genes in a more discontinuous or sharply bordered pattern. And this leads to brain arealization. So a similar theme, this is pretty similar to the one I touched on earlier, except we're seeing it here with actual gene expression. And we do know of some genes in the human brain that coordinate this organization, but it's generally pretty difficult to study this in humans for obvious reasons. But that's changed somewhat with the introduction of the Allen Human Brain Atlas, which I'm sure most of you know quite well. The dataset features 6 brains, from which 3,700 tissue samples have been extracted with really excellent brain coverage. And each tissue sample has undergone microarray gene expression analysis of 59,000 probes targeting 20,000 genes. And importantly for our purposes here, each sample also includes stereotaxic coordinates expressing the approximate location of the extracted tissue. So this allows for hypotheses to be tested concerning how gene expression is associated with or influences spatial organization of different brain structures. So I'm going to talk about 2 studies from our lab that takes advantage of this information. And a good place to start would be the hippocampus. So it's a relatively small structure, but it's one where we can see a lot of these ideas being represented. So most of us are aware that the hippocampus can be parcellated into several subfields, and these are kind of the regions of the hippocampus, if you will. And aside from being distinct in their cytoarchitecture and their connectivity, the subfields also show a fairly divergent pattern of gene expression that lets you differentiate them. But gradients can be observed in the hippocampus as well. And in particular, there's well-documented evidence of behavioral domain specificity and connectivity patterns varying systematically along the longitudinal or the anterior-posterior axis of the hippocampus. And this has been demonstrated quite elegantly in humans by several labs. However, the same longitudinal organization has also been shown in mice and other rodents, where different investigators have studied how transcriptomic patterns influence this functional organization, finding contributions from both gradient-like expression as well as sort of regional subfield expression. But similar studies were lacking in humans, so we set out to map a transcriptomic gradient mirroring the functional gradient along the longitudinal axis of the hippocampus in humans. So we first identified we wanted to sort of deal with the curvature of the hippocampus. So we identified a hippocampus skeleton, and we also identified 170 tissue samples from the Allen Brain Atlas that were in the hippocampus. And we found the closest skeleton coordinate for each one of these samples. So the goal was to have a single coordinate indicating where along the longitudinal axis of the hippocampus the sample fell. So we trained a machine learning algorithm to predict this location, so sort of where the sample lies along this anterior-posterior axis using just the transcriptomic information. So using cross-validation, we found that our model could predict the location of a sample along the longitudinal axis of the hippocampus with pretty high accuracy and pretty low error using just this gene expression information. And this perhaps indicates the presence of a consistent molecular gradient underlying the aforementioned functional specificity along this axis. And using a leave one subfield out framework, we importantly found that these results were actually fairly consistent across and therefore probably orthogonal to the hippocampal subfields. And similarly, we found leaving a leave one donor out analysis found pretty consistent results across the different donors. So to ensure these results were not limited just to the Allen Brain Atlas, we took advantage of a subset of the BrainSpan dataset, which includes RNA-seq data from laser microdissected samples from the brains of 4 prenatal human donors. And there were no coordinates, but we could use the precise anatomical labels to differentiate samples dissected from the posterior versus the anterior hippocampus. And so by applying our model to these data, we found we were able to discriminate the anterior from posterior hippocampus quite well. And that indicates not only that this model generalizes outside of the Allen Brain Atlas, but also that this genomic gradient appears to be active already during fetal development. So next, we were interested in how this gene expression gradient might influence relationships between the hippocampus and the rest of the brain. So we took non-hippocampus samples and we fed them to our model. We basically asked, I know you're not in the hippocampus, but if you were, based on your gene expression pattern, where along the longitudinal axis would you fall? And we kind of use this information to create what we call haggis, which is an index where higher values represent greater transcriptomic similarity to the anterior hippocampus, and lower values mean greater similarity to the posterior hippocampus. And when we looked across the whole brain, we saw this interesting pattern where brainstem and more anterior-ventral portions of the brain appeared to have a transcriptomic signature that was more similar to the anterior hippocampus, while the more posterior dorsal and brainstem and cerebellar sites were more similar to the posterior hippocampus. And we found that this haggis gradient, which is shown smoothed here at the top, was informative with regard to several cortical hippocampal relationships of interest. For example, we found a region's connectivity or structural covariance to different parts of the hippocampus were correlated with how similar its transcriptomic signature was to the hippocampus. And similarly, we found that this signature discriminated regions more vulnerable to Alzheimer's disease from those regions more vulnerable to frontotemporal dementia. So pretty interesting stuff. And we also performed a cognitive meta-analysis using Neurosynth and identified brain networks that not only shared similar expression of this hippocampal gradient, genomic gradient, but also which were involved in disparate cognitive or behavioral tasks. And these results corroborated really our expectations with regard to domain specificity of the different sort of poles of the hippocampus. So we started in the hippocampus, but we sort of ended up with this large gradient across the brain that seemed to influence or relate to 2 dissociated functional brain systems. And it got us thinking whether this could be considered the other way around, whether this hippocampal gradient might just be the local expression of a common brain-wide gradient. So that sort of influenced our thinking in the next study, where we zoomed out from the hippocampus and started looking across the whole brain. So in this study, we took a pretty similar approach, but instead of looking at just the hippocampus, we looked at tissue samples across the whole brain. And instead of constraining space to a single dimension, we actually looked across all 3 spatial dimensions. So for this study, we used cross-decomposition. We passed the gene expression information for a given sample to the X matrix and 3D spatial coordinates to the Y matrix. And we were hoping that the covariance of these factors might identify genomic gradients in the adult human brain. So we split our data into a training and test set. And in the training set, we used cross-validation to identify that partial least squares regression with the maximum number of components was the best model. So we also saw that all 3 of the components that we extracted explained variance in the spatial data far greater than expected by chance. And in our left out test set, we used the PLS model to predict the 3-dimensional spatial location of these unseen samples based just on the transcriptomic information. And here you can see the predicted values of the Y, Z, and X coordinates from top to bottom for each sample. And we found the model predicted location very well in this unseen data along the rostral caudal and dorsal ventral axes in particular, and maybe a bit less well along the medial lateral axis at the bottom. And to kind of visualize this, here we plot the Y and Z coordinates of each sample in the Allen Brain Atlas, and you can see the different brain regions are colored differently. And here's the predicted location of each of those samples with the same coloration. So the predicted version doesn't really look like a brain, but the spatial relationship between the major substructures is more or less preserved. And here we're just visualizing the model error, where samples in red had the worst prediction. And you can see the model seemed to perform the worst in parietal and prefrontal regions. But we were satisfied enough with this to move forward with characterizing the spatial and genomic qualities of these PLS covariates. So here's the first component. Okay, so the genomic expression of this component is visualized on the bottom, and the covariant linear gradient through space is shown on top. And we're seeing a transcriptomic signature that has sort of a ventral to dorsal emanation, and it seems to separate on the genomic side the cortex from subcortical structures. But we felt that perhaps a more elegant description of this gradient might be that the component differentiated brain regions based on their rostrocaudal position in the developing human brain. So as you can see here, component 1 expression is lowest in regions that originate in the telencephalon, and they increase as one moves caudally along the neural tube to regions originating in the met and myelencephalon. So once again, we wanted to replicate this data from another source, and we once again turned to the BrainSpan data set. So BrainSpan has great cortical coverage as well, but only has a few cerebellar samples outside of that. So here I'm showing how cortical and cerebellar samples are strongly dissociated by component 1 in the Allen Brain Atlas data set. And we replicated the same finding in BrainSpan with just the cerebellar and cortical samples. And furthermore, the BrainSpan samples were extracted from donors across the lifespan from sort of prenatal development all the way through adulthood. And we wanted to visualize how this relationship with component 1 evolves along the course of development. So here you can see that the samples were arranged from earliest age at 8 post-conception weeks all the way through to oldest age, which is 40 years. And we found that component 1 began to differentiate these structures already during the fetal period of development and plateaued in early childhood. So we also defined a second component that was characterized by this sort of posterior to anterior radiation, and it really seems very reminiscent of the hippocampus-based gradient that we found in the first study. But based on our conjecture from component 1, we wondered if this gradient might be important for regulating instead the dorsal ventral axis of the developing brain. And in each segment of the developing brain, we separated regions based on whether they originated in the dorsal or ventral part, and found that component 2 expression was consistently higher in the dorsally originating regions. And in the telencephalon, we found a specific pattern of expression that was lowest in the subcortex and increased as one moved from the frontal lobe and a posterior direction along the cortex. So once again, we wanted to replicate this in BrainSpan. And for component 2, recall that the expression in the cortex, which is where we had most of the coverage in BrainSpan, differentiated limbic from anterior from posterior cortical structures. So as such, we calculated the mean component 2 expression in several regions in the Allen Brain Atlas and correlated this with the mean component 2 expression of the same regions in BrainSpan. And we found a pretty remarkable correlation between these two data sets shown here. And this, not only indicates the generalizability of component 2, but again, we can see that component 2 expression began differentiating structures prenatally. And interestingly, in this case, though, the process seemed to continue through into adulthood. So it might be a little bit more dynamic. So finally, the last thing we did was, we were interested in whether these whole brain gradients might intersect and interact to promote cortical arealization. So we took the spatial gradients of the first 2 components and the third component, which I didn't have time to discuss here, and we just projected them across the brain to see where they overlapped. Pretty simple. So here I'm showing regions that shared concentrations of these different gradients. And, this is fairly speculative exercise, but if one squints, maybe you can make out some recognizable anatomical divisions of the cortex, see the parietal lobe or occipital lobe, and so forth. And so this is pretty interesting to us. And if you repeat the same exercise using instead the genomic components of the PLS covariates, which might be more relevant, this time the gradients define more of a sensory fugal axis. So you can see the primary sensory structures are differentiated from the sensory structures, the sensory support structures, which were then differentiated from heteromotor cortex and limbic structures. So all in all, in these 2 studies, we identified patterns of gene expression that occurred systematically along spatial gradients in the brain, both locally and more globally. The components were reproducible across and within cohorts, and they appeared to emerge prenatally. So while it requires further follow-up work, we are interested in understanding, testing whether and how these gradients may be involved in cortical arealization. So the first study has been published recently in Nature Communications. If you want more information, that also includes all the code that we use to do all the analyses and make all the plots. And if you'd like to learn more about the second study, it'll be presented at OHBM on June 30th at 7 p.m. Eastern time in the US, I think around midnight UK time, or you could just find me during the poster sessions. So with that, I'd like to thank my co-authors, our funding bodies, particularly the many amazing data sets and tools that made this research possible, and thank you for your attention.